Arias from Rice University, rising senior.
All right. Hello, everybody. Thank you, Dr. Ellen Bogan, for that introduction. My name is Ariah Richards. I am a senior at Rice University studying kinesiology and biochemistry. And this summer, I've been in the Ramirez lab, characterizing the swallow breathing coordination in male MECP2 knockout mice in comparison with wild types. So I want you all to think about the last time you drank a glass of water. Did you have to think about breathing while you were drinking that glass of water? The likely answer is not, because for most of us, our brains naturally coordinate this breathing, swallowing behavior. But that is not the case for individuals with different neural disorders, including disorders such as Rett syndrome. Rett syndrome is a neurodevelopmental disorder that is rare and commonly found in females. Approximately 60% of these individuals with Rett syndrome have a mutation on their methyl CpG binding protein 2 found on the X chromosome, which is why it's commonly in females. Symptoms include adolescent neurological decline, seizures, and for my project, we're really focusing in on this difficulty breathing and swallowing dysfunctions. So far for Rett syndrome, breathing is well characterized as dysfunctions and erratic breathing, breath holding, due to deficiencies in neurotransmitters such as GABA and serotonin. But what's less characterized is the swallowing behavior. So in this diagram, I know there's a lot, but really what we're looking at is that this is what it normally looks like for a human to swallow, and this is what it looks like when you're feeding a child with Rett syndrome, which is very irregular. This also usually leads to the children needing trach tubes and GT tubes due to dysphagia. So we really want to find out what's happening and what's disrupting. So my hypothesis is that male MECP2 knockouts will experience difficulties coordinating breathing while drinking in comparison to wild types. To study this, I used a behavioral study on 5-week-old MECP2 knockouts and wild types. We used male mice because their disease happened at an earlier time point, and for this project, we needed something a little earlier. The mice were placed in whole body polysomograph chambers. So here we see our little mice friend. They have an air exchange with oxygen, and so that we can monitor their breathing. And then there's a tube that feeds them sucrose, and a touch sensor here so that we know when they are drinking. And then we have a camera so that we can go back and look at our data that was collected. The mice were conditioned for 2 days, and then we looked at them every day consecutively because the onset of the disease can happen at a variable time point. So first, just some Rett syndrome characteristics that we notice in our knockouts. They weighed less than our wild types. They tended to have altered tongue control. And due to this altered tongue control, we tended to see them kind of drink from the side of their mouth. So they'd like turn their head to the side and try to like lick and appear to be struggling to drink water. They also experienced ataxia and hand wringing, which is when they're on their hind legs, they'll shake their arms really fast, which is a characteristic that we sometimes see in individuals with Rett syndrome. So moving on to our drinking data, we noticed a couple trends, although not significant, they are interesting. So our total lick duration was longer for knockouts as well as the time spent drinking, but the number of lick bouts was about the same. And then also the time to initiate. So the time it took the mice to find the water spout tended to be a lot more varied in knockouts and longer. If you look at the volume that the mice drank across the span of our days in the study, the amount of water they consumed between both groups was about the same, but over different lick durations, which we can appreciate in this graph where we see that the knockouts took significantly more time to drink the same amount of water. We then change our focus to the breathing behavior, we do notice an abnormal respiration between both groups. So here we see breathing traces for the wild type in blue and for the knockouts in purple next to their lick traces. And you can appreciate that the wild types have a fast and consistent breathing pace that slows a bit while they're drinking, but that is not the case for the knockouts. The knockouts' breathing is very irregular and becomes more irregular while they're drinking water. So now if we combine these two ideas of breathing and drinking, we get their breath-swallow coordination. And so here's a graph of their respiratory rate with frequency and breaths per minute. If we focus into just while the two groups are breathing, we see that the knockouts have a significantly lower respiratory rate. We see a similar trend if we're just focusing into while they are drinking. Again, the knockouts have a lower respiratory rate. But what's really interesting to note is that between the state of breathing and drinking, the knockouts have a lower respiratory rate that is significant, and this is not the case for their wild type counterparts. If we then focus in on airway protective behaviors, here we have a video that's going to show of an aspiration reflex after a mouse has drank water. You see this really big spike, which is the aspiration reflex. And as the video plays, we see difficulty breathing with breath holds, very irregular. In the video, it appears that the mouth is like coughing or choking. And then it starts to recover its breathing back to something that's more normal for them until it is completely recovered. It is interesting to note that even after the mouse experiences this, they will go back and drink again and cough again and aspirate again. So they are not deterred. They will still try to drink water. So in conclusion, we do notice difficulties in coordinating this breath and drinking in our knockouts. They take longer to drink the same amount of water. They have a low respiratory rate while drinking in comparison to breathing. They also have a lower respiratory rate breathing and drinking in comparison to their wild type counterparts, and they may experience airway protective behaviors in the form of aspiration. So in conclusion, I would like to acknowledge my lab, the Ramirez lab, my two mentors, Zach and Elisa, and my partner in this project, another undergrad, Miriam. The Ramirez lab has been really welcoming to me, and it's been an awesome summer. I'd also like to thank the NSSSP program, Dr. Ellen Bogan, Mrs. Ellen Bogan, everyone who has contributed to the program and made the summer a great time, and then also my fellow scholars. Y'all are great. And yeah, that's it.